안녕하세요. 네, 10시가 되었으므로, 시간이 되었으므로 오늘 행사를 시작하도록 하겠습니다. 안녕하세요. 2025년 제1회 학습과학연구소 세미나에 오신 여러분들을 환영합니다. 저는 오늘 행사 진행을 맡은 학습과학연구소 교수 이진경이고요. 오늘 세미나는 교육 난제를 해결하기 위해서 학습에 관한 융복합적 접근을 추구하는 학습과학연구소에서 사람의 두뇌가 어떻게 학습하는지 관심 있는 연구자들 및 학생분들을 위해서 마련된 자리로 오늘 이곳 12동 401호와 온라인으로 동시에 진행될 예정입니다. 오늘 학습과학연구소 세미나 자료는 여기에서 다운받으실 수 있는데요. QR코드를 스캔하시거나 또는 이제 인터넷 주소 링크를 통해서 다운로드 받으실 수 있겠습니다. 오늘 세미나 주제는 두뇌 기반 학습 연구의 기초, EEG와 ERP 연구 방법입니다. 먼저 오늘 세미나를 진행해 주실 연사인 홍콩대학교 이현경 박사님을 소개해 드리도록 하겠습니다. 이현경 박사님은 홍콩대에서 심리학 및 상담학 전공으로 학사를, 언어 및 청각학부에서 석사를 받으셨고 스피치 랭귀지 앤 리딩 랩에서 박사과정을 마치셨습니다. 현재 홍콩대학교 언어학과에서 박사 후 연구원으로 현재 홍콩대학교 언어학과에서 박사 후 연구원으로 주요 연구 관심사는 모국어와 제2언어 학습자의 언어 처리에 대한 신경 메커니즘을 연구하는 것인데요. 특별히 이중언어 사용자와 특수교육이 필요한 개인의 언어학습 및 발달을 조사하는데 오늘 세미나에서 얘기해 주실 EEG를 활용하여서 연구하고 계십니다. 박사님께서 사실은 중학생 때부터 중국에서 국제학교를 다니면서 이제 외국에서 오랫동안 공부를 하셔가지고 한국어보다 영어가 오히려 더 수월하신 분이세요. 그래서 글로벌하게 이제 우수하신 박사님을 모시고 오늘 강연을 듣게 되었는데요. 그래서 오늘 세미나 진행은 영어로 진행이 될 예정입니다. 포스터 제목이 한국어여가지고 혹시나 한국어로 진행될 것을 기대하셨던 분들 이제 혹시나 당황하지 마십사 미리 말씀드리고, 혹시 질의응답도 한국어로 해주셔도 물론 저희가 충분히 이제 의사소통하는데 어려움은 없지만 영어로 혹시 가능하시면 또 그렇게 남겨주셔도 도움이 될 것 같습니다. 자, 그러면 홍콩대학교 이현경 박사님을 큰 박수로 모시겠습니다.
So, hi everyone. Thanks for coming to this seminar. It's an honor to be here and to give the seminar to you today. So today the seminar will consist of three main parts. So the first will be introducing the EEG method. So I'll go over like basic principles and mechanisms, also the background of EEG, and also the advantages and disadvantages. And the second main part will be how this ERP method could be applied in research. So I'll introduce this ERP method, its main principles, as well as how we could define and characterize ERP components. And after that, I'll also go through some ERP components and like introduce some studies that use some ERP components. And then after then, in the third part, I'll also go through how we could acquire and analyze the EEG signals. So for example, how we could set up and record the EEG signals, and also how we could pre-process and analyze the data briefly. So first part, so what is EEG? Why are people interested in EEG? So you might have seen these wiggly lines before, and maybe you might have been a participant before in an EEG study wearing this funny looking cap. So we collect these EEG signals that are naturally occurring from the brain so that we could map this into some cognitive functioning, cognitive processes, and their underlying mechanisms. So essentially, EEG is a technique that records the naturally occurring electrical activity from the brain. So it's recording spontaneous and continuous signals like this. And this is the array of the electrode cap. So you can see a lot of channels that are placed on the scalp. So each line represents this channel. So each channel is recording this this wiggly lines, basically. So how does this work? So you can see this is the EEG electrode that's on the scalp, and this is the scalp. So this is the surface of the head. So and these are the neurons that fire. So you can see that until we could reach the signals emitted from the neurons, there are many layers of the head. So which means to be able to collect the EEG signals from this electrode, this signal needs to be large enough. To have the large enough signals, which means we have to have a large populations of neurons that act in a synchronized manner, like act similarly. So when a large populations of neurons are acting similarly together, then this electrical activity can be collected at this level of electrode outside of the brain. And these electrical signals are known to be modulated by a lot of internal states and external tasks. So for example, they're sensitive to your internal states, like your current emotions or your different levels of arousal. And also when you're performing different cognitive tasks, the EEG signals will reflect the changes. So I'll also share a brief background of EEG, like a brief history of EEG. So it's been like more than 100 years of EEG research. So in 1875, an English researcher named Richard Caden first discovered that the brain shows electrical current. And after then, a Polish researcher continued this research from like animals. And then in 1912, there was a Russian researcher that first did like animal EEG study. And then in 1924, which was exactly almost like 100 years ago from now, which is like 2025, this German researcher first recorded the first like human EEG. So it was the first time ever that humans recorded the EEG signals from human brain. So as you can see, like he modified some radio equipment like this to amplify the brain signals so that he can record the EEG signals on the graph paper. So if you compare this first EEG equipment from 1924, and this is the modern EEG system, so you can see the similarities. So for example, we are collecting the EEG signals from the brain, and then we'll use some sort of equipment to amplify the small brain signals, and then we'll like present it in a way that could be visualized. So why people are interested in EEG? What's good about it? So first, it has very high temporal resolution, which means it provides rich time-sensitive temporal neurodynamic information. So if you blink your eyes, that's about like 1 second. And then 1 second consists of 1,000 milliseconds. So the EEG technique can record information at millisecond levels. So you can see we can collect a lot of informations that happen in a very brief second of moment. And it's also compared to other equipment like fMRI is relatively cheaper. So to do a project, you need like EEG equipment, EEG caps, and different like supplies that are needed to set up the equipment every time. So it's relatively cheaper to maintain, like build and maintain the equipment. So there's an advantage. Also, if you're interested in auditory stimuli processing, it's relatively silent compared to other equipment. So yeah, so it'll be better to record clear signals. And also a good advantage is it's safe and have minimal risks. So it's non-invasive, which means like when you do an experiment, you don't have to like cut open your brain to collect the EEG signals. So participants will wear the EEG cap and will just collect the signals that are naturally occurring. So it's very safe. So therefore it's very suitable for children and clinical populations. Although when children see the equipment and the cap and the setup, like they'll be scared. And then they're like, what are you gonna do to my brain? Even for the parents, when they visit, they're like giving a suspicious look, like, what are you gonna do to my child's brain? But it's totally non-invasive. So we cannot do anything to the brain, even though we wanted to. So we can just collect the signals that are naturally occurring, just picking up the signals. So it's very safe. And so because of these advantages, it has like wide applications to different fields like clinical use. So in the hospitals, they would use the EEG to collect and compare EEG signals, usually for like brain-related impairments like epilepsy, dementia, and brain injuries with humans. And for research use, like many various fields like cognitive psychology, neurolinguistics. So for example, if you want to compare the cognitive processing or social processing of typically developing children versus children with special educational needs. So for example, if you want to compare the cognitive processing, for example, like inhibition of children with autism or ADHD versus typically developing children, you could use EEG to see if they vary or they are different in terms of the group performance. And also if you're interested in like linguistics, for example, if you're interested in certain language processing of monolinguals or bilinguals or across different languages, you can also compare the electrical signals of the two groups. And also you can compare the developmental trajectories, like for example, like you can compare the EEG signals of a developed like normal adults versus like children, like typically developing children at different age ranges, like elementary school, middle school, high school. So you can compare the development of the brain and their cognitive and social skills. And then like if you're interested in like emotion processing, for example, saying like the difference between like angry, happy faces, or like if you're interested in your arousal when you're seeing emotional faces like angry, surprise, neutral faces, you can also use EEG to compare the brain signals of these. Then what are the disadvantages? It's more like the concerns of EEGs that you need to be cautious about despite of all these advantages. So I just mentioned that EEG has very good time-sensitive like resolution. So to compensate for that, it has lower spatial resolution. So which means like EEG is very ideal if you're interested in what time, like when a certain processing occurs. If that is your research question, it is a very good technique. But if you're interested in where in the brain is this processing located, because it has poorer spatial resolution, it is not the best technique to use if you're interested in the source of this processing. And then another disadvantage could be the setup time can be long. So earlier you saw like a cap with like many channels. So if you're using many channels to do the data collection, each channel needs setup. So the setup time could be long. And then imagine if you're working with children who are like impatient or don't want or already a little bit scared of this lab environment, then it could be a little bit of challenge. So in this case, like you can use like less channels or use like different types of cap that could take less time to set up. And then another point to be concerned is that the EEG signals are very sensitive to artifacts, which means it's very sensitive to movements. So for example, like if you look left, right, your eyes are moving, right? So the EEG signals will collect the signals like this, which is definitely not the signal you want, right? And then, for example, if you're like nodding or chewing or like moving head slightly, the cab will collect those movements. So you have to provide very clear instructions for the participants that movement is very bad for us. So please sit still. And then also it's sensitive to like eye blinks and electrical noise. For example, if you have your phone or other electrical device near the EEG setup, it could also influence the signals. Yes, but like you can control this, like you can handle this. For example, like if you pay more cautions during the setup and also use like advanced filtering techniques, you can also correct this like artifacts to a certain extent. But of course, it's like better to collect like clear signal from the beginning. So then I'll move on to the second part, which is about applying this ERP method in research. So so you've seen this diagram, like the participant will sit in front of the simulation computer. So this is the computer that will present the task. So for example, if you're doing a task, the participant will be seated in front of the task and do the task while collecting these EEG signals from this EEG cap. And then these signals are large enough, but still very, very small. So we need this like amplifier to make the signals large enough so that it will be visible in the digitization computer that we'll see. And then the stimulation computer will communicate with this digitization computer that will show this electrical signals. So for example, these are the signals from these like respective electrodes. And then the stimulation computer, for example, when you see a happy face, you'll set a code like number 1. And then when you're seeing an angry face, it'll show a trigger like number 2, for example. Then this electrical, this waveforms, it'll mark the condition on the waveform at the same time, so that later you can just, you can refer to this and analyze the signals. So for example, this is condition X and this is condition O. So when the recording is being recorded, we will mark this trigger for each condition when the participant is viewing the signals. And then we will cut out the signals and then average them. So here you saw the cut out signals for X's and O's. And then in the end, for ERPs, we'll average all the X's and average all the O's. So although like each cropped part will look very wiggly and like nothing like what is it, but if they're averaged out, hopefully it will show very clear patterns. So essentially, ERPs are the electrical like potentials that are time locked to specific events. So those events could be like sensory, motor, or like cognitive events. So whatever like processing that you are interested in. So because they are like time locked to a specific events, it's easy to interpret this processing that I'm looking at, that this one is actually reflecting the specific cognitive processes that I'm interested in. So, so there are like different categories of the ERP components. So usually the early components like P1 and N1, those are sensitive to like physical properties of the sensory stimuli. So for example, like light. So they are happening at like around like 100 or before like 200 milliseconds of time, which is like very, very brief and usually at the like subconscious level. And then after then, like around from like 300 and onwards, there will be like later components, like endogenous components. So that's when we say actually the cognitive processing are picked up and start. So these components will reflect the information processing and whatever that's modulated by the task. So here you see like different electrodes. So each channel will be representing like many, many different lines. So like there's so many lines. And how do we know, for example, the P1 component, which is related to visual processing, is? So if I see all these channels, how do I know which one is P1? Is it this, 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 or this? So to know, like we consider like several like elements of the ERP components, like polarity, latency, and scope distributions and functional associations. So first, the polarity is whether it's negative or positive. So on the scale, you see minus is up and then plus is down, right? And then it's pretty like simple. So P1 component, for example, is positive, and N1 or N2, they're negative. But one thing to bear in mind is that like when it exists in the positive or negative range, it's not about that. So for example, like N2 is technically having like positive value here, right? So it's not about the absolute value, it's more about the trend of the peak. So for example, P1 peak here is like positive going, right? So that's why it's a positive component. For N1 is negative going, same as N2, although it's at the positive range, it's like negative going peak. So that's how we name the ERP components based on the polarity. And next is latency. So latency means like when does the peak occur? So for example, like ERP components like P1 is around 100 milliseconds. Like P2 will be around 200 milliseconds. And P300 will be around like 300 milliseconds, and 400 will be around 400 milliseconds. So it's easy to map based on the latency. So latency means like where the peak is. So you see for this graph, there is a peak here. So from 0, like when does the peak occur? So that's about the peak latency for naming the ERP components. And the next is like electros. So when you read the studies, you'll find the trend that like, for example, like for an ERP component, they'll use and report like similar array of electrodes. So that's also important. And then next is the topographical scope distribution. So which means like where in the surface of the brain the activation is strong. So for example, P1, you can see is at the back of the head, like right side and back of the head, like occipital right hemisphere region. And for N1, you can see it's more like bilateral, which means both like left and right of the back of the head. And for P1, so this is nose, which means it's at the top of the head, like frontal central. So each component will have different like distributions. So we also need to check that. And then last is the functional associations. So which means what kind of cognitive functions are traditionally or consistently associated with this component. So for example, if you see this like peak in this time range, like is it related to word processing? Or like when you see like changes like deviance detection or face processing. So in the literature, we need to consider like what function, what mechanisms are reported to be associated with this component. So we need to consider all these elements together because sometimes on ERP, like some ERP components happen at similar time, like 200, 250, but they could happen at like frontal or occipital, like different scope distribution, and they could mean like different cognitive processes. So it's important to consider all these factors when you're like determining or like reading EG or ERP related literature. So I'll like talk about like a few ERP components. So to like go over the elements that I discussed just now. So this is a component named N170. So you can see here that polarity is negative. Like in different studies, it's all going negative, like negative going peak consistently. And then for the latency, well, the name says N170, so the peak is around this time range. So you can see all these peak happen between like 150 to 200 milliseconds in different studies. And then the scalp distributions. So this component is known to occur mostly at the back of the head. You can see this like parietal, parieto-occipital regions in the channel array. So you can see these studies are consistently reporting like P8, which is here, and PO8, which is like here, and then also these like occipital like brain regions. And then also for the functional association, like it's known to be sensitive to face processing. So you can see that there's like more negativity for faces compared to when you view houses. Very distinct, very like different, right? And then when you see face versus cause, you can see very different negativity, which is going more negative. And also for upright and inverted faces, because they're faces, they're both showing like N170, but for inverted faces, it will show like enhanced negativity because like you'll use more cognitive efforts to process like faces that are a little like different. So this is another study that used like N170 that I did before. So using this knowledge that N170 can tell you about face processing, you could use it to compare like this face processing mechanisms across different groups. So for example, like children with ASD or autism, they're known to have like reduced social interest and different like face processing strategies. So in this case, so I compared the ASD children and TD children for viewing real and cartoon faces. So there are like real faces of like Hong Kong children at between like 9 to 12 years age range. So they're about like elementary school students. And then also I created like cartoon faces that are created based on the real faces. So as you can see, the blue is like typically developing normal children, and the red is the autistic ASD children. So you can see that compared to the TD children who showed enhanced negativity, enhanced N170 negativity, for the red, which is the autistic children, they showed reduced activation. And it was also like left lateralized, which means this difference was prominent on the left hemisphere compared to the right hemisphere. So from this information, we can interpret these findings that autistic children compared to TD children, they show reduced real face processing. And interestingly, this difference was only observed in the real faces. So for example, if you see like cartoon face processing of the two groups, they show similar trend. So the group difference was only observed in the real faces, but not the cartoon faces. And another component to share is the N2, so which is a component that typically marks and indexes like cognitive inhibition, which occurs around like 200 to 300 milliseconds. So to measure this inhibition, like a typical task that researchers use will be like a Stroop task that compares between like congruent and incongruent condition. So by meaning like congruent is like, it's red in text and red color, so they match, right? So they're congruent. But incongruent is like the text is red, but the color is blue. So there's some discrepancy, like difference. So that's what it means by incongruent. So, and another task is like a flanker task. So you can see congruent, like the arrows are like consistent, like all same arrows, compared to incongruent condition, that's like in the middle, the arrows are different. So you measure if there's any difference between the congruent and incongruent like conditions using this N2 component. And then another popular paradigm is a go, no go task. So you'll be presented with a series of go tasks. So for go, you'll like respond, like click, 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 click. And then there will be a few no-go trials that you're supposed to not respond. So imagine if you have to keep on responding and suddenly have to stop, it'll be difficult, right? So this is what we mean by like your ability to inhibit. So we'll measure how efficient you are or how much more cognitive efforts you need to use to inhibit yourself. So N2 at the negative range around 200, and then usually it'll be placed around like frontocentral, which is around here of the brain, of the scalp. And then you can see that compared to the go trials that are pretty like flat, the no-go trials are very negative. So there is a clear difference between the go, no-go, go and no-go trials. So in one of my studies, like I did like emotional phase go no-go task, which means the different phases will act as go and no-go trials. So for example, in this block, happy faces were go. So when you look at, when you see happy faces, you'll click, click, click until you see other emotions, for example, like angry or neutral or surprise. If it's not happy, you're supposed to like stop yourself and not respond. So that would be the challenge. So from this, you can see the blue is typically developing children. So for go trials, there was no difference, which is expected. But for no-go trials, you see that the red, which is like autistic children, is clearly more negative compared to the blue. So this difference only happened at the no-go trials, which means for autistic children, like it takes a lot more cognitive efforts to inhibit yourself compared to the TD children. So now I'll move on to like how we could acquire the EEG signals and like possibly analyze them. So you've seen this figure. So before all these happens, like we need to conduct some various like steps to set up the EEG cap. Well, I see some of you have done it already. They're like, yeah, it takes long. So first, like you need to clean some of the face areas because like you'll be wearing the EEG cap, but besides on that, you want to attach some like little electrodes on your face. So for example, if you attach them like here next to the eyes, here, it will collect the signals of like left and right movement. And then if you place them above and below your eyes like this, like it will collect eye blinks. So it's important to attach this eye channel so that later when you analyze the data, you could correct these artifacts. So before placing these electrodes, like we want to clean the face areas so that, for example, like dust, like human faces also could have like oils. So we want to clean the face so that the electrical signal will be very clear. And then everyone has different head size, right? And then different head shapes. So for example, some people will have wider forehead. Some people will have like narrow forehead. Some people will have round head. Some people will have like flat head. So we want to place the cap in a way that could accurately or close to accurately represent the array of each person's head. So to do that, we need to measure the head size to first know which cap to use. Like children will use very small caps. Adults will use like usually medium. Some people need large, some people need small. And then also people have different like lengths of here. So, and also here. So we'll all consider all these factors so that we could accurately place the cap. And then the next step is what might take a bit of time. So you will put a cap on a human participant's head, right? Then you see a lot of channels on the cap, which means each channel needs set up. So this display, this program shows how each channel is being set up. Basically, if it's red, it's not ready, like very not ready. And then if it's like blue, close to blue, it's like okay. So usually the norm is like 5. You see like this F5 is like 1.8, which is below, so it's very good. And some of them were like 100, above 250, like 17.6. So to reach that number, we'll use like this conductive gel. So this gel helps to enhance the connectivity between the gel and the signals. So we will use this like syringes, like, and then needles. So it's not like a very sharp needle. So they're like very blunt, so you can touch and it's not sharp at all. But like just the presentation of like syringes and needles could freak people out. So you need to like explain to them, especially if they're like children or parents who are already like a bit scared and worried, like that you can let them touch the needles that is not sharp at all, and then it won't do any harm to you. So you can demonstrate that. So usually you put the gels in the syringes, and then you will like put the gels in each channel until you can reach the ideal signal. So I'll show you an example video. Okay. So so this is a video that I made when I was doing like EEG experiments like a few years ago. So it can give you some ideas about like how the setup can be. So in my old lab, we had like a hair washing chair, like a salon. So like it's optional, but like if you wash your hair before the experiment, the signals could be quite better. It could improve the quality of the signals, because like even though you washed your hair last night, like overnight, it could collect dust, oil, and many other things. So it's the best that your participants wash the hair in the morning, or at least like in front of my eyes. So that's the most ideal way. So we would usually like mandate that participants wash their hair in our lab.
Second,
Like
children
this.
will wash hair on our hair washing chair, blow
And
dry
then
hair, and comb hair.
And then it was one of our protocols that they also comb their hair so that it could remove any more dirty things that could possibly be on your head, so that like we were doing every little steps to improve the signals. Well, but yeah, children didn't like them so much. It was mostly for us.
Parents will be helping with the procedure to make children comfortable. Next, we'll use
And then here you see I'm like wiping the face areas.
alcohol swabs to clean some face areas where we'll place electrodes on. We'll measure
And
the
I'm
head
measuring the
size,
head size,
put on the
and
EEG
then
cap.
placing this EEG cap on the children. And then here you see there are like some electrodes that were not placed on the face area yet.
Cap of the suitable size,
And then adjusting
and
cap,
affix the electrodes to
then-
the face areas
Yeah.
that we cleaned earlier.
and then placing these electrodes on the face like this. So yeah, that's me a few years ago.
We'll squirt a gel through.
So after then, this is the, the syringe and the gels. So you see there are a lot of channels. So I'm adding the gels and then
the holes in the cap to increase the
and
contact.
shaking a little bit so that the gels will sit on the scalp very well. So when you add the gels, like, of course you have hair too, right? So you need to add the gels wisely, because if you put too much gels, like the channels are quite close nearby. So if you put too much, they will connect and then they'll act as one electrode, which is not what you want. So you will want to put like little bit of gel and then shake so that they will spread well and sit on the scalp well. But like this shaking part could be a bit challenging because like no one wants something on their head, like touching like bit sharp thing. So yes.
between the electrodes and the head.
So the challenge is to finish the setup time in a very efficient manner and in a manner that the participant doesn't feel very uncomfortable.
To apply the gel in these small holes, we'll use a flat-tipped syringe. The gel is easily washed out of hair, and
After the
children will
task
wash
is
hair
finished,
upon the
they'll wash the
completion
hand.
of the
It's easy
test.
to remove.
After
And during the
applying
task, when all these channels are finished set up, the participant will perform the task in front of the stimulation computer. Is this? So in the old lab, we would have the stimulation computer in the sound booth. And then the booth is also electrically shielded. So in outside of this room, like just outside, there will be like two computers that are the same as this one, and also the like EEG recording computer, so that we could see them together on top of a small monitor that we could monitor the children because we are not with them physically, right? So when the children is like moving, then we could stop the experiment and then talk to the children. Hi, do you need a break? Do you mind sitting down? Something like that. So we can communicate with the child, even for the adults as well. I mean, it's not necessary that adults are better. Like I've had some adults falling asleep during the EEG experiment. And back then I didn't have camera, so I wouldn't know what's going on. Like outside of the room, I'm just seeing the EEG signals that are weird. Like when someone's very sleepy, like there's like signals very wavy, like instant noodle like signals. We can see, this person's very quiet, very sleepy. But like, and then like if we do not see any response, which is like recorded by using the markers from the stimulation computer, then we know something's wrong. So in that case, we'll see. But if you have like a monitor to like see the participant inside the room, is like not a big issue.
the gel, children will start computerized tests.
Yes. So that was like the procedure we did. Okay. So like how is this recorded? We saw the stimulation computer will send the triggers to the digitization computer, which will be shown in the computer like those marker codes. And what's next? So the next important thing is the cap. So as I mentioned, there are like many channels, but there are different kinds of caps too. So the most like common, I should say, the electrode will be like 64 channels. Like if you read the literature, like 64 channel is usually the typical ones that researchers use. There are also 128, which is like so many channels that I've only seen like one or two people actually using them and taking like so long to set up. But like it really depends on your like research questions. So in a lot of experiments, you don't need a lot of channels. So you wanna cut the time so that the participants will not be too tired or bored and fall asleep during the actual experiment. You wanna cut the time. So like if your like experiment does not require a lot of channels, then you could also use like 32 channels as well, which is also perfectly fine. And then there are like other kinds of caps. They're similar, just like different materials. Like the right one is like a silicone, so it'll be easier to dry. And then there are other types of systems. So the ones we saw just now are like the electrodes are mounted, like attached to the cap like this. So you just wear them and the electrodes are there and you adjust to each electrode. And there are other kinds of caps too, like you wear this like hard hat with holes and then you will mount the electrodes yourself. So they are like both like used like commonly. And then like we'll amplify the signals. So like the most common ones are the non-portable ones, which means the amplifier are like physically in the lab and you cannot move them. So you will bring the participants and then set up there and then do the experiment, which is very typical. So like these are like some popular devices. And there are also other labs. Many labs also use like portable EEG. So which means the amplifier that we saw earlier, like this, are reduced in size. Like it's very compact. It's like this big. So you can move the amplifier, which means you can do the experiment like anywhere. So like it's really depending on your research question and also your lab space. For example, if you do not have like a EEG lab, like you do not have lab space, like you have to set up each time, borrow a space and do the EEG experiment, which can be used for many other experiments, then you could consider using like portable EEG so that whenever you have EEG experiment, you can bring these devices and do the experiment. So now like we have these signals that need to be like processed. So for data treatment, it's popular to use like MATLAB with like different toolboxes like EEG lab, ERP lab, field trip. Like those are like different like toolboxes that you can use to process the signals. So like, for example, like you can use like GUI based like toolboxes. So this is like EEG lab and ERP lab. So GUI based, so there are like buttons and there are like different like pop-ups that you can see what's happening, like what am I clicking, like what analysis I'm doing. You can use the program to just click and like type. So it's very user-friendly. But like many people also use like script-based analysis. So it's basically you have like MATLAB and then you just like write and modify a bunch of codes so that you can do the same kind of analysis. So like this might look a little intimidating if you have no like coding like background, but like there are a lot of sources on the internet. And also like if you want to do more like advanced level of analysis, like it gives a lot more like freedom and flexibility. So yeah, so many people just choose to do like script-based like analysis, including myself. And some people use like a mix of these two. So like you can like, if you're new, you can first start with like GUI based analysis and then get like more familiar, then and then you can move on to the script based or like mix them. So this is the raw data you collected. Then like there are like several like pre-processing steps that need to be done that I'll just briefly explain. So first, like I mentioned that the data is like at millisecond level. So it has very rich information and the data file size is huge. But like for kind of like processing, like you don't need that big file size. So first we'll like resample the data, usually to like 250 is usually enough for most of the analysis. Then we'll filter the data. So we'll use different filtering techniques so that we'll remove some very high pitch frequencies or very low pitch frequencies so that the data will be filtered. And then you see compared to raw, which is like blue, it'll be like a little more smooth. And then after doing some initial filtering, like you'll do some like artifact correction using like ICA analysis, which is independent component analysis. So it'll give you some pop-ups with different like brain components. So from here, you're trying to identify what are related, possibly related to like artifacts. So for example, some of them could be representing like brain movement or eye blinks or like head movement, something like that. So you will identify from here and then correct them. So when you like correct them, then you can see compared to the blue lines, the red line is the corrected signals. So for example, you see there was like big like increase or drop in the signals for the blue, which was like corrected after this like step. And then after like re-referencing the data, as I mentioned, we'll like epoch the data, which means like crop out and cut the data based on each condition. And then after cleaning more, we'll average the signals and then use them to analyze the data. So for example, we can plot the ERPs. So like it's really up to you. Like if you want to first see the whole head, like how these signals are represented in like each channel, you can see all the electrodes or like selected electrodes that you're interested in and compare. And then, for example, like you can compare the red versus black, green versus black, which are like different conditions, and then see how they're different in terms of the peak and latency and the amplitude. And then you can also make some like difference maps. For example, you can subtract the baseline and the red condition within the given time range and see how big is the difference. So you can color code these differences so that it will be easier to see from the graphs. And then you can also make like time course topographs. So for example, like how this color changes or this processing changes with time. So for example, it's from like 310 milliseconds till 610 milliseconds. So for each condition, you can see there are like different trends or different patterns in this like processing. So after plotting, like based on this information, you can also measure or extract the amplitudes and latency. For example, like this is the peak, and then this is the peak amplitude. And this is the mean amplitude, which is like a popular way to average the signal. So within this time range, you will like get each value and then average the values to get the mean amplitude. And then the latency, like how long it takes to have this clear peak. So we'll get like numerical numbers for these like amplitudes and latencies and run like statistical tests. Like you could do like correlation, ANOVA, and curves, and like linear mixed effect models. Like different ways of like analyzing this signals and numbers. So that, for example, this peak of the red, whether this is faster, whether this occurs faster than this black one, or whether this one is significantly more reduced compared to the black one. So using these tests, we can compare like, we can do like significance tests, whether these differences or these color differences are significant, so that we can interpret the result and like say that, oh, actually these two conditions or these two groups were meaningfully different. So yeah, so like concluding, so like wrapping up the seminar. So EEG is useful. So it provides very rich information in the neurotemporal dynamics, like at like milliseconds, which cannot be done by like other like techniques like fMRI. But of course, like EEG data is very sensitive to like artifacts and noise. So we need to use like different filtering or data processing like techniques so that we can handle and correct these artifacts and noise so that possibly we could save more data and also design the experiments in an appropriate way. So for example, like even for adults, like if you need to sit wearing a cap in a room doing tasks for very long, like you want to move, right? So we need to design in a way that we give like room like a time for the participants to move. So that will like give them some time to relax and not be overly too tense, and like so that they can take like breaks at like the appropriate time and then focus back to the task. And same for children, like they need breaks very often because like usually their like attention spans like 2 to 3 minutes or like much less if you do a task with them. So yeah, we need to provide like enough breaks, and also like add some like safety measure during the experiment. Like if the child is like, I want to stand up, I want to move, or like accidentally moving, you can pause the experiment, and then in that way you can save more trials. And then also like, of course, like you will need to give clear instructions to the participants. Some like it could make a difference. For example, like if the participants knows that like moving now will not be good for the researchers, then they will like pay more attention and try not to move so much. So that's important for like our research. And then like once you acquire some like basics skills of the like ERP, like you could also like move on like in the future to do like other kinds of analysis, like time frequency analysis, like looking at like power differences and like frequency differences, and like connectivity analysis. And then you could also extract some like entropies or like different features from the EEG signals, and then use them to do like machine learning models, and then possibly use these like classification features to classify, for example, like whether these features help to distinguish between the typically developing children versus autism children, or like typically developing children versus like typically developed adults. So we could like use that in the future. So that's it for the seminar. Yes. Yeah. So I'm open to like any questions if you have.
Hello, thank you so much for your talk. It was really great to see EEG and ERP analysis methods. I have perhaps it's a naive question about ERP analysis. You mentioned that the go, no-go task can be differentiated with positive and negative trends, as well as the amplitude differences. And I was wondering if those differences are comparing only between, for instance, go or no-go trials for only trials in which participants correctly responded, or whether it's just regardless of the response, just all trials are compared.
Yes. So usually like when we do like analysis, usually we'll compare like only the correct trials. So usually we'll exclude the incorrect trials and just focus on the correct trials, because we think that accurately represents the processing that you are actually doing and what I'm interested in, actually. So like for other types of like task paradigms or like processing, we would usually focus on the correct trials. So that's why like when you plan the experiment, it's important to have like enough number of trials. So the norm is like usually we want at least like 40 trials that are safe to use. So which means like you need to consider that there will be some trials that the participants didn't respond correctly. So those will be gone, and participants will move or have some artifacts or noises. So some trials will be gone too. So you need to, when you plan the experiment, like you have to have very enough number of trials so that you'll have very clear and robust like peaks and signals as well. Yeah.
Hello, I am in the major of educational technology, and thank you so much for your presentation. And it was a great lecture that helped me get a better grasp of EEG. And for novice like myself who might want to study EEG further after today's presentation, could you recommend any books or paper or other resources for learning about EEG analysis? Thank you.
Yes, actually, there's like a very classic book that all the novice or like new to ERP will EG or ERP will use. There's like an introduction to EG/ERP methods by Locke. I think the LUCK. The first one is like usually like is published in like 2005. So it's like a classic textbook that people use when they are first, like they're new to EG and ERP. I mean, myself also read that book when I was first like studying EG. That book like shows all the basic principles. And I mean, today I only introduced like a few components, but they have more comprehensive like discussion of the ERP components and also analysis. So if you're interested, and that book will be also like a good start. And then also just now I was showing like EG and ERP like toolboxes, like their websites also have like a good like resources, like the explanations. For example, if you are interested in the GUI, then they'll have some like screenshots of like what to click basically and what to do. And then like their websites and other like websites will also have provide some like sample codes that you can like play around with. Yeah.
Thank you. Thank you very much.
Thank you for your presentation. I got some detailed information from your presentation. I have one question that, so you mentioned EEG has lower spatial resolution, but I'm a little bit confused because it has many electrodes and we can see all signals of them. So why is it has lower spatial resolution? So yeah.
I see. So yeah, that's a good question. So if you wear the cap, and then I mean, I was visually showing like diagrams like where, right? But as I mentioned, it's the sculpt distribution. So it's around the where on the head surface, the activations are strong. So it's slightly different from the source. So when we are talking about the spatial resolution, it's more about the locus, like where of the brain area the processing comes from. So for example, like our brain is round, and then like different brain parts are like some are like close to the surface, but some are like below. So with EEG, it's like very difficult to capture that. So we know around where, like briefly it occurs, but we don't know exact point, like exact location of it. So if you're interested in that, that's more for fMRI. Like they don't provide information much about the time, but they will give like more precise information about the location. Does it answer your question?
Thank you for your insightful lecture. So I wonder, actually, I could find you analyzed by millisecond unit event and event response. So I wonder, what would be the appropriate event time and event unit or event response unit per for your good EEG measurement?
Oh, you mean like which time frame I need to
time
cut
frame
out?
or time, the analyzed unit
Analyze unit like
or
millisecond or
appropriate event time. Like, because very short event response you analyze.
So so for example, like if you're interested in the ERP component that occurs at around like 400 milliseconds, so it really depends on the ERP component, then you will like crop out around 400 milliseconds of the continuous EEG signals to analyze that component. And if you're, for example, interested in like N2, which is around 200 and 300 millisecond, then you will cut out like that area. Are you asking about that, or are you asking about like how long each trial should be?
Yeah, how long or how many times in one research?
Oh, so usually it really depends on your like paradigm, but like if it's like a typical like short one, like for example like a word or like faces, then usually we'll crop out like minus 200, which is like 200 milliseconds before the presentation of the trial, like of the visual stimulus or auditory stimulus, and then until like 1000 millisecond. So we usually like do like initial cutting of that much like time range until like 1000 millisecond. Then like from then we'll extract the values of the time frame that you are more interested in. So if you're talking about like epoching at the beginning during the pre-processing, usually you do like 1000 millisecond, that's like a norm. And then depending on your stimulus, for example, if you're doing like sentences that are like longer, then of course there should be like a longer time frame.
Thank you for the lectures. I want to wonder that wearing the EEG hat may affect the progress of the experience, because some child can think that the hat is maybe weird or strange, and I think that could affect the experience, especially in the case of children with some disabilities, maybe autistic. The difference can be worse than the general children. So does this affect the experience? If so, how do you remove this effect?
Okay, yeah, thank you for your question. So actually that is like a very good question, because I worked with like quite a lot of children with autism. And then like, you know, like for autistic children, they're very sensitive to sensory input, like touching. So not everyone, but some of them especially dislike having the cap. So like when you're setting up, like you will, like I usually let them touch the cap so that they're initially comfortable with the cap. And then this also like the size of the cap could matter as well. So like if the cap is like too tight, the uncomfortableness will be like larger. So you will measure the head and then give the appropriate head size cap. And then also after wearing the cap, like if you adjust them, like it's less comfortable, but still like there's individual differences. So we will like communicate with the child and then ask the child if they're okay to proceed. So like we will not proceed if they're like, oh, I hate this, like I cannot do this, I want to take it off immediately. We wouldn't like proceed. But like from my experience, like most of the children, like there was no child that was like, I will not do it, let me go. Like no one just like took off the cap and left. So it's it could be uncomfortable, but like it's the level of discomfort that everyone could bear with. So usually that was not a big issue.
Thank you for the interesting talk. I have one simple question about the number of participants for doing this kind of research, because the EEG signal is rather noisy. I believe that the number of students would be large to get enough the reliability. So usually how many participants are necessary for doing this kind of research?
Right. So like it's for the standard in the EEG studies is slightly different from the behavioral studies. So usually the rule of thumb, like the normal, like the typical number will be like 30. So usually when we see like 30 participants, then we'll be like, oh, it's like enough, unless you have like so many conditions. So the rule of thumb is like 30 participants for like each group. And then like, of course, but like you want to have like good 30 participants. So usually we would recruit like a few more than the 30 participants, so that we would have at least 30 participants. So there will be like usually like enough number that you can see like clear signals. So like if you have like 5 participants or something, usually you cannot really see much because of the noise. But from like 15 onwards, you can usually see like a general trend of what your research or findings will be like. So like usually when you reach like 15 participants, it's like a good time to do the like pilot analysis to see like whether you've been collecting the right results. And then like when you reach 30, you could do like a real analysis, which would be like a enough number usually. Yeah.
Okay, so how many the autism children you had in your research you mentioned?
So for my study, for one of my studies, I had like around 50. So actually, it was a little unnecessary. So I didn't have to have that much. It's because like we were doing, so because this experiment was like a part of like another large project. So we had a lot of children. So we had to test them as the part of the project. But usually when I do like other studies that like I'm currently analyzing, like they're all around like 30.
Okay, thank you.
The first question is about the research example or research trend in learning sciences using EEG or ERP. Could you tell?
So like I briefly discussed about like my research and like other research briefly. So like what I was specializing in was like children with special educational needs. So like now it's also a trend that like you compare the EEG signals of like the typically developing children versus like children with special educational needs, like children with like learning difficulties or reading comprehension difficulties like dyslexia, or children with like attention like problems like ADHD, or also like children with autism, like who usually have like impairments with like inhibition and also like social functioning. So you could like compare like across group like measures to compare across the groups. And then like to do that, usually you would use like, you still use a lot of the ERP like methods like I presented today, but now people are also like employing like time frequency analysis like this, and then moving on to like connectivity analysis, which usually you need like more channels, like at least like 64 or more, or like using these to run like machine learning models. So those are like the trends that's like happening and receiving more attention these days.
대답이 되셨기를 바랍니다. The second question is on whether blood flow over skill or blood pressure change affect EEG signals in terms of electronic conductivity. Are those factors considered and filtered within the processing procedure?
So usually not much of the blood pressure, but some people also consider like cardiac changes. So like the heart rate, like if it's like a big heart rate, it could influence the electrical signals. So earlier I showed like during the pre-processing, like we could do ICA component analysis. So it shows in the component analysis, like some components that are related to cardiac, like heart rate movement. So in that case, we could also like correct them during the pre-processing.
And the third question is related to the previous question, I think. Yes. So could you tell us more details when we are conducting a research for children with autism using EEG or ERP method?
Yeah, I think it was like kind of addressed already, but yeah, so like we know like the big deficits or impairments that autistic children have, or like in their like deficits in like executive function, like inhibition or like cognitive flexibility. So we could use like ERP like components that are related to those like cognitive functions and compare with the typically developing children. Or you could like observe the changes. For example, if you're providing like intervention to the autistic children, and then like you can do like longitudinal studies or like collect EEG signals at different time frames across the intervention, then you can compare the changes and then like whether the intervention is working. So that's one way in terms of the cognition. And also like autistic children show like notable like differences in like social emotional processing. So that's also another huge area that people are researching about in terms of like ASD research.
In terms of the next question, maybe I need to ask not this question first, and then we need to go to this question. So compared to fMRI and MEG, using EEG, is this more popular or less popular?
Well, in terms of popular or less popular, it really depends on the research question. I mean, both are very popular, but not all the labs have fMRI equipment, and not all of them have access to fMRI equipment, and they're very, very expensive. Usually, like a single lab doesn't own fMRI equipment. So in terms of accessing the equipment, like fMRI is slightly at a disadvantage unless you have like a collaboration with the hospital. But like, so in terms of accessibility, EEG has definitely more advantage. But both like these two techniques are tackling different research questions. So if you're interested in the time-related changes, how this processing goes on, then EEG is more ideal. But if you're interested in like which part of the brain this processing happens, then fMRI is more ideal. So it really depends on like what you are looking for and what you want to do in terms of your research.
Thank you. Actually, this question, the exact question is a kind of question with some opinion. Well, the asker think that fMRI and MEG can overcome the difficulties of or the limitation of EEG. Actually, fMRI and MEG has some advantage to have better visuality, but maybe these methods have some disadvantage. Are these methods less used because of the cost? That is the question.
So I think I've already addressed that question. Yeah. So it really depends on your like research goals. So like even though it might have like slightly limited access, like if you're really interested in like source localization, source analysis, but your primary goal is not about time, like when this processing occurs, then you should like it's more right to go for the fMRI, like, but like unless like it's not about like accessibility, it's more about like what you're trying to do in terms of the research aim.
Thank you. And could you explain in more detail the criteria for removing noise artifacts such as muscle or eye blinks? And is there a risk of accidentally deleting meaningful data during the removing?
Yes, totally. There is 100% risk that you might delete like the very good data that you actually need when you already do not have a lot of data. So that's why we have like many pre-processing steps, and then in between each step, you want to go back to your data set and check whether you overcorrected the artifacts. So for example, if you are too strict in terms of artifact correction or removal, and then from like ICA component analysis earlier, if you like select a lot of them, like then there is a high chance that you are removing a lot of the useful information that didn't have to be corrected. So we call it like overcorrecting. So in terms of like like ICA component analysis for detecting the eye blinks, like muscle artifacts, like movements, it's better to be more conservative, like less strict, and then so that you can save more data set and do not overcorrect the data, which is like an incorrect representation of like actual reality, right? So you usually like we recommend taking more a conservative approach in terms of pre-processing and like artifact removal. Then we'll use more like advanced filtering techniques that's like automated, like based on like algorithms to clean out more noises. Yeah.
And you said each have weakness in spatial resolution over MRI, but do you think the approach to source level using analysis tools is helpful in paper publishing?
So yes, so like not in a very, very recent times, but like there was like a trend that researchers using EEG were combining like some like toolboxes that could like do some source analysis like S-Loretta, like yeah, they were trying to use those toolboxes to find or add more insights about the source analysis. But now in this like recent trends that is actually it's not like a recent trend that you're trying to do this anymore in terms of doing like a Loretta analysis to know more about the source. So yeah, so instead, like if you're really interested in like source level, then you could like combine fMRI and EEG, like do both, and then like analyze and correlate them together. That is that could be more ideal instead of trying to add more like source level information from the EEG, because like EEG signals already do not have good spatial resolution, so it may not provide like accurate representation of where the source is actually. But yeah, there's researchers still using these techniques, still trying to add more about the spatial. So like you can try, but like in terms of paper publishing, like I'm not sure, because if you use those, then like reviewers will definitely comment that you are trying to do something spatial with EEG. So, so if you're considering adding this for the paper publishing, then maybe that's not the best idea. But if you're interested, then like, of course you can try.
And this participant is interested about your research. So in terms of your research, is this a clinical trial now, or is it for just the academic purpose?
Yeah, so mine was not a clinical trial. So it was more of like a research project. So like I was just like collecting the EEG signals during cognitive and social tasks instead of like clinically meaningful like task. So yeah, so you're right about it was like a university research project.
And the next question is there an easier way than EEG to measure brain activity in the classroom rather than in a lab?
Yes, so I think EEG is already kind of quite easy, but like you can consider using like portable. So for example, if you want to collect like brain activity in a classroom setting, instead of bringing the participant to your lab, then definitely it should be like a portable EEG. Then, and then like depending on your research questions, like if you're not interested in, if your research does not require a lot of channels, then you can use like 32 or even less. Some people use even less channels, then it will take much less time to set up. So in a classroom setting, like that could be better.
The next question is, considering the complexity of EEG research, such as removing noise and ensuring clear data, even for 30 participants, this person thinks that data analysis alone would take a significant amount of time. So considering the duration varies depending on the study, how long does it typically take for your research team to conduct EEG research project?
Okay, so let me think. So like it depends on like how many tasks you have, for example. So each experiment, like including the setup time, usually it will be at least like 2 hours or a bit less if you're doing like a very short task. Like if your task is about like 30 minutes up to like an hour, then like the EEG session will take about like 2 to 3 hours, which is like one morning or one afternoon session. So based on that, you can plan like how many bookings you can make to the EEG lab. For example, if you have only one EEG equipment, then like if you do 2 a day, how many can you do in a month, right? So like doing the experiment itself will take some time. And then usually like the data analysis, like you will do it offline. So meaning after you collect a bunch of like EEG data, then like when you have some, then you will analyze them together. So yeah, pre-processing will take time, and then plotting and extracting like values and running statistical models will take time too. It really depends on like how many conditions or how many like aims, goals you're trying to address from the research. But yeah, it'll take months.
Thank you. The next question is about the unique contribution of EEG or ERP, especially for developing individualized or tailored teaching strategy or learning lessons.
Yeah, I think it was also kind of addressed earlier. So like, for example, like if you're interested in like special educational needs children, then you could definitely compare the like brain, like a cognitive or social processing of those children versus like typically developing children. Or also you could compare like different groups of participants, different groups of children to see the improvement as they age. So yeah, so from comparing these EEG signals, you can get some like interpretation or insights about what these children are lacking and what's the underlying reason, like what's the cognitive mechanisms, like a real cognitive mechanisms that they're lacking. For example, like if they have like some deficits and like processing social stimuli, is it from like early processing when they perceive the faces, like are they different from then, or is it the later processing of the faces, like emotional or face related features, like the later processing that's different? So from then we can like gain insights and possibly like apply this to like educational research.
Thank you. And in addition to N170 and N2, there is another component such as P300. Are there general trends in ERP analysis across industries using?
So if I understand you correctly, like, do you mean if there are like general trends in ERP components that are like commonly used across industries? So, okay. Oh, same person, right? Yeah. So, yeah. So, well, it's a bit difficult to say across industries. I'm not sure what specific industries you're talking about, but maybe you're meaning if there are like general like ERP components that are like popularly used. So So yeah, you mentioned P300. That's also a very popular component. For example, like if you're studying like cognitive inhibition, like P300 is another like very popular component in addition to N2 that people usually investigate. Usually they investigate N2 and P3 like together. So like N2 is about like earlier cognitive inhibition, whereas like P300 is more about like a motor related like inhibition. So they happen at like a different time frame and then slightly different mechanisms that that translate over time. So yeah, so and then like so it really depends on like the field and the research. So like if you're interested in like phase or like word processing, like N170 or like LPP, which is like late positive potential, which is about like emotion processing. And then if you're interested in like word or sentence meaning processing, then you would usually do like N400. N400 is another like very popular ERP component that you can see a lot in the literature.
Thank you. Maybe this one is from the same participant. So 혹시 이 질문하신 분 이 바디 무브먼트가 피지컬 바디 무브먼트 말씀하시는 거 맞으시지요? 네. When conducting experience using EEG and ERP, is it possible to have a lot of body movement by the participant? And is it okay to use the data in this collected in this situation?
So the EEG and ERP I've been showing, like they cannot have like a lot of body movements because the EEG signals are very, very sensitive to like artifacts and movements. But I know like there are some studies that use like portable EEG with like very limited channels that record signals during movement, but their mechanism is different. So for example, this one is like a time lock signals that at viewing or like accessing the certain stimuli, we are seeing the event like potentials, but those are more about instead of ERP, you would just look at like power or like frequency information, so that is slightly like freer from movement. But yeah, generally, usually we would not want a lot of body movement. Yeah.
So to be specific, for example, is it possible to use EEG or ERP while the participant is playing the piano or playing the musical instrument? Is it possible?
Usually we don't do that because that will definitely affect the signals. Yeah,
I'm sorry to hear that.
Yeah, we usually don't want any movement.
Then how about this? When two experimental participants playing a musical instrument in a different way. Yeah, the same question. So then how about this? If one practices, one read the musical score while the other participants look at the video, other people are playing the musical instrument. So one is reading the musical score and the other one watch the video. In that case, is it okay to use EEG or ERP to compare the differences between these?
Yeah, so if you're viewing like videos, yeah, it is okay. Yeah, there are a lot of studies that involve like video, like presentation. Yeah, and then like analyze the signals that are evoked by different components of the videos. For example, like you could compare like a very peaceful video versus like emotional like videos, and then compare whether like the signals like induced by different like emotional valence or different like components of the video induce different levels of arousal in the EEG signals. Yeah.
대답이 되셨기를 바랍니다. The next question is about research for children with autism. So in your research, is there any case seeing the similar pattern between children with autism versus children without autism when you use EEG or ERP?
Yeah, of course. So autism, like, you know, like the full name is like autism, like spectrum disorder, which means like the population of children with autism is not a single group that shows similar trend. So some children, like, they're very similar to typically developing children. They're like very high functioning, like some of them are even better than typically developing children in terms of some like cognitive task. So like for the children that I assess with EEG, I know that they have autism because we only invited those who have like formal diagnosis. So they definitely have autism. But in terms of like EEG and brain patterns, sometimes they do not show like much difference compared to the typically developing children. So yes, so yes, so even though they show similar patterns, like the group consists of like very high functioning, mid functioning, low functioning, like everyone's very different. There's like a very like each individual is very different. So like clinically you could still like say that they still have like autism, even though in the brain patterns the difference is not significant.
Thank you. Next question is, if one continually observed their own brain activity through EEG, it seems that they may develop a visual awareness of which brain regions are activated during certain activities. If that is the case, this awareness of brain activity might influence EEG signals. In the field of sports, directly observing, stimulating, and understanding one's muscle movement help activate those muscles more effectively. In some cases, muscles that were previously unresponsive may start to show noticeable movement. So could gaining a deeper understanding of brain activity help individuals stimulate and enhance their own neural activation? In other words, does learning about brain activity contribute to enhancing a learner's cognitive abilities?
See, so like, do you mean like having the awareness that you are being recorded of the brain activity? Like, could this enhance someone's cognitive abilities? Or like whether you know like explicit and implicit way of measuring the paradigm influences the cognitive abilities?
혹시 질문해 주신 분 대답해 주실 수 있을까요?
Sorry, you said you're shy, but sorry, I have to pick you on for the clarification. Hope you don't mind too much.
네, 한국어로 편히 말씀해 주셔도
네.
됩니다. 네.
네. 어느 부분이 활성화되는지에 대해서 이해하는 것이, 그러니까 나의 뇌가 어떻게 작동하고 있다라는 것을 인지하는 것이 학습 능력에 도움이 된다라고 한다면, 그럼 우리가 무엇을 가르칠 때 그것이 우리 두뇌의 어느 부분에서 학습이 되고 어떻게 작동한다라는 것까지 학습시키면 그것이 교육에 도움이 되겠느냐. 그러니까 두뇌 기반 학습 연구의 기초가 오늘 제목이니까요. 그래서 우리의 뇌 구조에 대해서 이해하는 것과 그것을 학습자에게 인지시키는 게 학습 능력 향상에 실제로 도움이 되느냐. 그러니까 뭐 가슴 근육이나 등 근육 중에 안 움직이던 근육들도 계속해서 자극하고 거울을 보고 하면은 그 안 움직이던 근육들도 사용할 수가 있게 되잖아요. 그러니까 작동하지 않는 뇌의 부위가 혹시 있어서 혹은 작동이 덜 된 부분이 있어서 그것을 활성화시키는데 뇌의 구조를 학습하는 것이 도움이 되느냐는 그런 질문이죠.
Yes, So it's a bit tricky because as I told you, all these processing that we are interested in happen at a very brief second. For example, as I told you, this is 1 second, but the components we are referring to are for example, 200 milliseconds. You wouldn't even know what's happening. So, so many paradigms and tasks happen at the subconscious level. So, in terms of when they're performing the task, it would be difficult for them to be aware of exact the collection or awareness of which component and which brain areas we are trying to collect. So we could map this after the experiment, when you're interpreting the results, so we can have insights about around which brain activations are important for this processing, and then we can use these findings for the educational research purposes. But in terms of whether knowing about the collection of the brain activity, it would be difficult for them to know. So it would be difficult to relate this to whether this will enhance the learner's cognitive abilities. Does it answer your question?
아까 발표에서 EEG 썼을 때 정확하게 어느 부분이 작동하는지 정확하게 측정은 어렵다는 단점이 있었던 거를 말씀하셨던 것 같아요. Yeah, could you answer the question? Using EEG or ERP, actually that helps understand the brain activity. Knowing our brain activity, can this help to improve the students' learning competency, actually?
In terms of the brain areas, don't think it's very relevant for the EEG technique if you're interested in that part. But with the findings, you can generalize later so that it could help with preparing the teaching materials, whether this way of teaching could improve the learning competency.
혹시 대답이 안 되셨으면 더 질문하셔도 되는데 괜찮을까요? 네, 그러면은 나중에 끝나고 더 이야기 나누는 걸로 하겠습니다. 혹시
Dyslexia.
그래서 홍콩대학교에서 아마 이현경 선생님이 있는 그 연구실에서 했던 연구 중에 하나가 난독증이 있는 학생들이 어떤 이 두뇌의 펑션이나 이런 것들에서 어떤 일반인과 차이가 있는지를 연구를 했고, 그런 연구에 기반해서 응용을 해서 어떤 학습을 돕기 위한 소프트웨어라든지 또 교육 방법을 개발한 걸로 알고 있어요. 그래서 그런 응용된 부분에 대해서 조금 더 설명을 해주면 좋을 것 같습니다.
So children with dyslexia, they can perfectly read, but in terms of comprehension and understanding the meanings, they have difficulties. So using EEG, you can present various different types of stimuli, and then you can examine which kind of patterns of the language they are having more challenges with. So for example, like stimuli or like this pattern of like teaching could help. Like it will be easier for the dyslexic children to understand. But in condition B, like dyslexic children have more difficulties, then you can compare this and then use this information to design the teaching materials that dyslexic children have less challenges with. And then so in some EEG studies, you can also find out like which strategies work better for the dyslexic children or even like autistic children for like face processing strategies. So in that case, you can use this information like what strategies are used by the dyslexic children that's different from the typically developing children. Then you could apply this knowledge for the educational research so that it could be easier for their learning and enhance their learning. Yeah.
네, 혹시 또 질문 있으실까요? 아, 네. 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 네, 편하신 대로.
제가 이해한 바로는 그 EEG 연구는 그 베이스라인과 그 자극 이후의 그 차이를 비교해서 이렇게 분석을 하는 것으로 이해했는데, 제가 궁금한 점은 예를 들면 인터벤션 스터디 같은 경우에 뭐 사전과 사후의 그 결과를 해석할 수 있는지, 예를 들면 뉴로플라스티시티 같은 거를 이게 베이스라인과 자극 이후의 흐름으로 파악하는 건 이해가 됐는데 사전과 사후의 결과를 통해서 해석을 할 수 있는지가 궁금합니다.
Yes, definitely. So that's also like a trend in the neuroscience research. So like usually when you do intervention, like you would have like behavioral findings, whether this is working, but it's only like a part of like information. So it's also like popular or more in trend nowadays. When you give like behavioral like interventions and assessments, you will also do EEG like assessment like before and possibly middle and also after to see whether there's indeed like neuroplasticity effect, whether like there's like overall changes in the brain signals in terms of the cognitive like functions that you're interested in and targeted by the intervention. Yeah, so yes,